Well, the topic I'm going to talk about today is microbial genomics, bioinformatics and antimicrobial resistance. If you want to pick up the slides, if you haven't got them already, there's a URL there which will allow you to download a copy of the slides. And what I'm going to do is provide initially an introduction to microbial genomics, bioinformatics and AMR, so that sets the scene. Then I'm going to take you through a case study on Mycobacterium tuberculosis, and a case study on Acinetobacter baumannii, and then finally a case study on the ICU microbiome. And interspersed with those case studies, there's going to be an opportunity for student participation. So when I used to do this in the days when there's only a couple of people in the class, we actually did it in the class. What I'm going to do now is when I get to those parts, I will pause, give you a chance to go and do the participation stuff, and at the very end of the talk I'll give you a bit of help in case you had any trouble with that. But the idea is that you're supposed to find your own way through those parts first as the default, and only if things get really difficult should you go and look at the crib that I give at the end. Just to introduce myself, my name's Mark Pallen. I've trained as a medic originally. I worked at Barts and then did a PhD where I led a university challenge team for Imperial College. I've had various chairs in the past. My research interests straddle across bioinformatics, genomic epidemiology, metagenomics and microbiomes. I'm the director of UEA's MRC-funded Microbes, Microbiomes and Bioinformatics DTP, and I'm also director of a MRC-funded cloud computing project called Climb Big Data. And you can find out more about me from those various links there. And you are presumably looking at this on YouTube on the channel there where I have put some of my lectures in the past. Now, as we get started, I'm just going to put some ideas in your head and let them work their way through as we're going. So you can think about this in the background as you're listening to me talking. So the first question is, how big is a bacterial genome? Have you got an idea roughly how big you expect that to be in terms of numbers of base pairs? How do you think bacterial genomes differ from the human genome sequence? What are the important differences there? And once we've got a bacterial genome sequence, what do you think it can teach us? What can we learn from that? And I'll point you here if you're interested in further reading about all this. It's a bit old now, this paper. It summarised 20 years of bacterial genome sequencing. Next year we'll be celebrating 30 years, but it does provide you with a broad overview. I'm going to quote some of the figures from that particular article as I go forward. Now, for me, the bacterial genomics golden era was that period from 1995 to 2007. So for most of you, probably the area in which you were born, or even some of it before you were born. But in this period, it was a heady time to be alive in science, because we got genome sequences from all the significant bacterial pathogens of humans, animals, plants. And this gave us tremendous new insights into pathogen biology and into the biology of microorganisms generally. So even for an organism like E. coli that people have been studying for the best part of a century, half of the genes were only discovered when we sequenced its genome. And for many other pathogens, many other organisms, much larger percentage of the genes became available only through genome sequencing. Second phase, we started getting multiple genomes from the same species, and we started to realise that there was tremendous genomic diversity even within the same species of bacterium. So this is just a timeline of that period. I'm not going to go through all of this in detail, but basically how did this first revolution come about? Because this guy, there's two people down there at the bottom there. Fred Sanger, who invented what we call Sanger sequencing, which allowed us to sequence short tracts of DNA. And then Craig Venter, who showed us that if we actually assembled all those short tracts of DNA together, we could actually create a whole genome sequence. So Fred Sanger himself had invented the idea of shotgun sequencing, but it was Fred Sanger and his colleague Ham Smith that made us realise that actually we could do this at scale with the right computer programs. And here you can see the various timelines. As I say, Fred Sanger invented what's known as Sanger sequencing of dideoxy chain terminate sequencing in 1977. It was 1995 that we got the first complete genome sequences of free living organisms, Haemophilus influenzae and Mycobacterium genitalium. And as I say, I'm not going to go through all of those, but you can see we get more and more multiple sequences. When we got more than one sequence of E. coli, we started to realise, wow, only a small fraction of the genes in E. coli are conserved in all strains. There's huge variation there. And then we started to get more difficult pathogens. Trophobema whippleyi was an organism we couldn't even grow in the lab in exanic culture, but we managed to culture enough of that in tissue culture and sequence it. Mycobacterium leprae was another one where they had to use a 9-banded armadillo to get enough DNA to get the first sequences. And as I say, it was a heady time to be alive for me because I was involved in some of these early bacterial genome sequences here. And in those days, these were getting into high impact journals because they were, they were the very earliest genome sequences, you might say, were over £1 million in cost. Certainly it was costing hundreds of thousands of pounds to get these initial genome sequences. And this is from a paper by David Relman and his colleagues where they reviewed what you could actually do with a bacterial genome sequence. So once you've got a bacterial genome sequence, you could look at the population structure and evolutionary history of the organism. You could look at outbreaks and strain detection. You could look at what's required for this thing to grow. You can reconstruct its metabolism. That's what happened with Trophobema whippleyi. You can develop diagnostic tools. You can take the proteins that you can see encoded in that genome and express them. You can then use those for drug design. And of course, you learn so much about the potential pathogenicity factors or virulence factors present in these genomes when you look at them. Of course, it's not always the things that you're looking for that turn out to be most exciting. There's lots of unplanned but important spin-offs. So one very early example was that genome sequencing project on Campylobacter jejuni, where what was discovered was a new glycosylation system that was much more similar to what we see in mammalian cells than had been seen previously in bacteria. And this led to a new vibrant programming of so-called glycoengineering, which allows you to stick glucose and other glycoconjugates onto vaccines. And it's a very vibrant area of research even to this day. Another interesting development as a bioinformatician of some note called Eugene Koonin at the National Centre for Bioinformatics Information in the US, he looked at loads of genomes and said, oh, there were these funny little repeats here and there were some genes associated with them. And he looked at those and found that there were bits of phage genomes mixed in, and he said, oh, this is probably some kind of immune system that targets specific sequences from bacteriophages. What was the great leap forward was that Jennifer Doudna and other colleagues realised that you could actually use these systems to cut DNA in a very precise way, and this led to genome engineering of humans, plants and animals. Probably an advance that counts as one of the greatest scientific breakthroughs of this millennium, in fact, and it is still, the ramifications of that are still being played out as we create new breeds of plants and animals, redoing agriculture, and also allow us to get involved in gene therapy. And another thing that's interesting that's happened is that by looking at all what's out there, you can treat these as kind of toolkits and use them to make new kinds of bacteria, new kinds of biology that aren't seen in nature. And Craig Venter, again, was one of the leaders in this field by actually creating the genome of a free living organism for the first time. He created a mycoplasma genome. This all changed from around 2007. Up until then, each genome project was still a major undertaking for a lab. But suddenly we put our foot on the accelerator. Things got 1,000 times faster and over 1,000 times cheaper. So this is what economists would call a disruptive technology. It was a game changer. And very quickly we actually ended up with what we call benchtop sequencing. So sequencing instruments had a form factor that meant they could just sit on the benchtop, and they were cheap enough that any kind of lab, every major department in a university, even a large research group, could afford its own sequencing instrument. And this was transformational. It basically democratized sequencing. So in the past, there would be major sequencing centers, like the cathedrals of sequencing. Now, it was everywhere across the country, hundreds and hundreds of groups doing this. The big change here was this idea of Illumina sequencing, where you, instead of cloning things in bacteria, you actually make your clones on a two-dimensional solid surface, and you do this through a process of chemistry, which is carefully done in a spatial orientation, so you can grow little molecular colonies to then sequence the molecules that gave rise to them. And this gave us all of this new approaches. So we started to be able to sequence outbreaks much more freely. We could get into ancient DNA. We could look at thousands of isolates and start to see huge new aspects of biology that we hadn't seen before. And so, as I say, fundamentally new approaches, this solid phase amplification of clonal templates in molecular colonies. And a massive increase in the number of clones that you get, if you like, the number of sequences you get from these sequencing reactions. The drawback was that the read lengths you get are somewhat shorter, initially quite a lot shorter than you got with traditional Sanger sequencing. So at its best, Sanger sequencing would give you perhaps a kilobase of sequence in a single read, whereas with this, started off at 50 or 100, 150. It got to be a few hundred as time went on, but nonetheless, it never quite got up to what we saw with Sanger sequencing at its best. And then a third revolution came along with the idea of single molecule sequencing. So instead of having to build little colonies of things, either in bacteria or on surfaces, you just took individual molecules and you sequenced them. One of these was part1 BIO, first of these really, a method of sequencing by synthesis with single molecule templates. Problem had low accuracy but long read lengths, and the other problem was that the instruments were huge. I mean, there was a video going around in the early days of a huge great truck having to deliver one of these instruments, and they had to smash the window in and take the window out of a lab and winch this thing in. Things have got a bit better since then. But a real game changer has been the so-called palm top sequencing from Oxford Nanopore, where you have these instruments that you could fit into a palm top there, into your hand, so-called nanopore sequencing, where a DNA molecule goes through a nanopore in a membrane, and you measure changes in the voltage as it goes across. And again, this is low accuracy but very long read lengths, and the accuracy has got better and better over time, so it is now at the stage where it is rivaling Illumina sequencing in terms of accuracy. Okay, that's just a brief introduction to the technologies. Let's go back to that question I asked you. What about the difference between a microbial genome and a human genome? Well, when we look at bacterial genomes in particular, bacteria and archaea, what we see are what we might call what you see is what you get genomes. So you'll see a protein coding gene encoded in the genome sitting there. It's all one continuous sequence. It starts at the start codon, ends at the stop codon, and that contrasts greatly with what you often see or generally see in the human genome, where genes are interrupted by introns, which mean that only 1% of the human genome is coding, whereas in a bacterial genome, 90% of the genome is actually coding for proteins. The intergenic regions are generally very short, there's very little repetitive or non-coding DNA. And introns do occur in some bacteria and in some bacteriophages and mobile elements, but they're like very, very rare actually in bacteria. And the protein coding genes are generally quite short. The kind of rule of thumb is they're about a kilobase in length. Just going back to the first line there, typically a bacterial genome would be in the megabase pair range, so 2 million base pairs, something like that as the average. They might range down to 500,000 up to 7, 8, 9 million, but it's all in that megabase. Whereas a human genome, we're talking gigabase pairs of sequences. Another key point is that the genes typically in organisms like E. coli, not always in all bacteria, but in many bacteria, they're arranged in what we call operons, where there's a single promoter just upstream of a series of genes arranged together in a row, and those are transcribed as one single transcript in response to common signals, and generally they encode genes that have similar functions. Fewer non-coding RNAs, that is one of those things that's changing because people are discovering more and more non-coding RNAs in bacteria, but compared to what you see in eukaryotes, this is still a fairly underdeveloped field. The other thing that you see in the human is there are many different transcripts, the splicing of introns and splicing variants and so forth. So what you see in the DNA and what you actually get in RNA and what you get in proteins don't have a quite straightforward kind of linear relationship that you see in bacteria. Bacterial genomes generally consist of a single chromosome, usually circular, but you can have some linear chromosomes, and you can have some organisms where you have multiple chromosomes. Vibrio cholera is an example there. We do also see these independent autonomous replicons, which can be DNA that can replicate in the cytoplasm independently of the chromosome, although sometimes they can just jump into the chromosome, integrate into it, and they can vary in their copy number. Sometimes there's one plasmid, one chromosome. Sometimes there are many, many, many copies of the plasmid compared to the chromosome. And in a sense, these are sort of your add-on, non-essential genes that give you a kind of, often called the accessory genome. They provide an adaptive advantage in certain conditions, but they're not required for survival in every condition that organism is going to encounter. Just one slide really, just to introduce the idea of microbial bioinformatics. And basically what happens here is we analyse all those sequences that we get from bacterial genomes. I'll say more about the kind of analyses we do in a moment. But one important kind of thing that we have to look at when we enter the field of microbial bioinformatics is that when you're doing this professionally and at speed and in earnest, use what we call the command line, and typically using what's called a Linux type operating system, which is a variation of an operating system called Unix, where you have to basically type in commands. It means it's harder to remember. You have to remember what those commands mean, but it's very powerful and versatile. Very easy to write code for. You can write Python scripts and so forth. Very easy to create pipelines. And this is where the cutting edge of bioinformatics is. You're probably more familiar with what we call GUIs, or graphical user interfaces, or web interface, which is much more intuitive, and the examples I'm going to give you for your hands-on work are actually going to be using the web here, but that's just a taster. And this is intuitive, easy to use, but less powerful, less versatility, much harder to write programs for pipelines, and there's always a lag time in development. So the best programs that use a GUI are always some way out of date in terms of what they can achieve compared to command line. And when you have a command line interface, you explicitly type commands and you have the Linux operating system execute them using what's known as a shell. That's a program that interprets the command we wish Linux to have executed. So typically you have a command, various options and arguments. So typically we might have a command like ls, which will list all the files in a particular directory, and we type that and Linux just does what we ask. One of the key points about bioinformatics is you can do it anywhere. You could, there are interfaces you can get on your mobile phone to give you a command line. You can certainly work from home doing this stuff. And here's quite a few years ago now, people were talking about an initiative called Code for Palestine, where people were actually doing useful bioinformatics in a conflict zone in Gaza. I'm afraid, given recent events, all of that has been blown out of the water because they don't even have internet or mobile signals for most of the time. But it is an example of how this is an extremely portable kind of approach that can be done anywhere, unlike lab work where you're actually having to be in the lab and hold a pipette. Just to give you a taster of some of the things that go on in the field of bioinformatics, one of the tasks that we have to look at is how do we assemble sequences. So as I said before, with Sanger sequencing, with Illumina sequencing, and even to some degree, less so with the long read sequencing methods like nanopore, you have to assemble all the bits you have into a larger sequence. And one of the problems you got here is if there are overlapping sequences, it's fairly straightforward. The ones at the bottom there, you can see how they overlap. Oh yeah, we can string those together okay. But the problem is where there's repetitive sequences, you don't know where that particular sequence read should be aligned, because the ones at the top there's two options here in this sequence. And so where do you put the alignment there? And this inability to work out how to handle repetitive sequences can be quite a setback. When we try and assemble a whole genome, what we do is we create what we call contigs, which are areas of contiguous sequence. Now, it may be typically what we do is we're sequencing from either end of a sequence, a piece of DNA, and we get a bit of sequence from either end. We can put all those together and we can assemble them and get overlapping sequence to make the contigs. Sometimes it may be that in our library we don't actually have a sequence from the middle of a region there, but we know that region's there, so we can still assemble things into sort of a large sequence map, if you like. Occasionally, it may be that there's a physical gap in that we just don't have any sequence representing a region. This used to be a problem when you were cloning in E. coli. If there were parts of the DNA that actually were lethal to E. coli, they couldn't be cloning E. coli if they had cloned something that was toxic to E. coli. We tend to see that less frequently now that we're using kind of chemical methods by physical methods. But even so, sometimes pieces of DNA might not get represented in your library. And so when we add all the contigs together, we get what we call scaffolds. And then we add all the scaffolds together, we get a genome assembly. Once we've got a genome assembly, what we try and do is annotate that. So you can't just look at a load of ACs, Ts and Gs and instantly interpret what it means. What we do here is we do a process called annotation. We add information about the particular sequence features to that, what we call a flat file of DNA code. One of the first things we try and do is look at the potential protein coding sequences. They're often abbreviated by the term CDS. And we can use what we call homology searches to predict function. So we can search a large database of all known protein sequences and look at, what other protein sequences look statistically similar to this one. And if they cross a certain threshold, we can say, they're likely to be doing a similar job. They're likely to be located in the same cellular compartment. They're likely to share a common ancestry. But we can annotate other features apart from protein coding genes, non-coding RNAs, promoters, repeat sequences, mobile genetic elements. One of the things that we often look for when we're looking at bacterial genomes is the origin of replication. We can locate that using various analyses. And we can just count the number of bases in a genome, the number of genes. One key feature that's often looked at is the G plus C percentage. So as G pairs with C and A with T, and vice versa, there's always going to be the same number of Gs in the genome as Cs in the genome, but there is a variation between the G plus C and A plus T ratio between different bacteria, and that can be a quick and easy thing to calculate, but actually quite informative. Another approach that we can do in addition to assembly is if we have a very closely related reference genome, we can map the sequences. We have the short reads. It tend maybe not working very well if we do what we call de novo assembly, where we just try and assemble them as like doing a jigsaw puzzle on its own. What we're doing instead when we map against a reference genome is like trying to assemble that jigsaw puzzle using the image on the lid of that box to tell you what it's supposed to look like. Oh, you know that there's that much sky there. Those bits, a deep blue sky, need to go together in that area. Lots of clues there. And the same applies when we do sequence mapping. And one of the things we can do when we're looking at closely related strains, when we've mapped sequences and seen what the differences are, is we can look at what we call SNPs, single nucleotide polymorphisms, sometimes called single nucleotide variants. And these basically are the smallest changes you can see between genomes. So just a single base pair change, as you can see in those two genomes. And these are useful in two ways. So if they're biologically significant, they can tell us, this organism is resistant to an antibiotic because it's got a change in an antibiotic resistance gene that renders it resistant. Maybe it's an enzyme and there's a subtle change to the active site that is enough to make it resistant. But there's also a huge number of SNPs that actually are neutral. Most of the changes when you make changes to DNA don't actually have any effect, partly because of the degeneracy of the genetic code. So you've got 4 codons that spell glycine. It doesn't really matter what you do to the third codon. It's not going to make any difference to any amino acid in a protein. And similarly, even in proteins, different amino acids can do pretty much the same job. So changing an alanine to a glycine or vice versa, probably not going to make much difference to your protein. But those neutral changes are actually very useful because they allow us to reconstruct the ancestry, the phylogeny, the family tree of the genomes by looking at how those different SNPs are related to each other and concatenating them and drawing trees based on that. Now, just a few words about antimicrobial resistance. So this is a problem that predates the medical use of antibiotics. So basically, the organisms that make antibiotics have to be resistant to their own antibiotics. But the spread of those resistances to other organisms became a big problem once we started using antibiotics. The increasing global availability of antibiotics, particularly since the 1950s, what we often see is uncontrolled sale, meaning that you can, in many parts of the world, you can just go and buy antibiotics over the counter and use them when they're not actually strictly indicated. If you looked at an expert and said, would you give antibiotics? They said, no, but they get used anyway. And many of the antibiotics we have a broad spectrum, and these are more likely to induce resistance than narrow spectrum antibiotics. Another problem is that antibiotics used in livestock for growth promotion, and to some degree in when you're growing plants as well. So sometimes apples get sprayed with antibiotics and so forth, and this selects for antibiotic resistance. And we're basically releasing large quantities into the environment. During manufacture, you go downstream of a factory that's making antibiotics and look at the water outflow. It'll be stack full of antibiotics. Similarly, a hospital is going to be putting lots of antibiotics out into its sewage. What's going on here is often called Darwin in the drugstore. So there's basically this evolutionary process of natural selection going on. So you start off with a population where, this is very figurative the way I'm showing it, you might have one resistant organism in billions and billions of cells, one resistant cell. But you then apply the antibiotic and you kill off all the sensitive organisms, But because of the way bacteria grow, and they'll grow, one cell becomes 1 billion cells within 24 hours of culture, you can turn the whole population resistant in a very quick time. So this is simply this process of natural selection. How that mutation actually happens, how do you get that change? There are two options. One is point mutations or mutations in the genome where bits of DNA change or are lost. The other thing that bacteria do is they engage in a kind of global group sex where we get horizontal gene transfer, and this also fuels the spread of antimicrobial resistance. And the problem we have is that this is not just an incidental problem out there in the wider picture. It's actually focused on bacteria that cause serious infections in humans, and that resistance is increasing in many bacteria. And problematic is the fact that infections are often caused by multi-drug resistant bacteria, where they acquire multiple resistances, not just one antibiotic resistance, but many. And we know that this causes excess morbidity, so more disease and excess mortality. More people die. And just for the bean counters, the penny counters, this also puts up the cost of healthcare. Patients stay in hospital longer. They're given more expensive antibiotics. They require longer periods of time on intensive care unit. So antimicrobial resistance is a bad thing in many, many ways. And here's just an example of how we get resistance and how it spreads. So animals get antibiotics and develop resistance, particularly in their guts. Those bacteria stay on the meat, and then if they're not cooked properly, they get into the human food chain. They can get into animal feces, and that's spread on to food crops and gets into the environment. A patient gets antibiotics, and the resistant bacteria in his gut there grow to large population sizes, and they can then spread in the community. If he's in a hospital or in a nursing home, you get even more of an enrichment of those resistant bacteria, and they spread around the hospital, and particularly between patients, direct transfer, but they can go onto surfaces in inanimate environment in the hospital, or they can be spread by healthcare workers. And this idea that there is this seamless transition here between what goes on in animals and humans and the environment is sometimes called the One Health approach, so that veterinary and human medicine both apply here. A few years ago now, 10 years ago in fact, the O'Neill report came out, and this looked at what the problem might be in years to come, and they O'Neill predicted that by 2050 antimicrobial resistance will be causing 10 million deaths per year, compared to currently where it's maybe 700,000. And so it actually become more of a problem than cancer. And he put forward a whole range of suggestions for the different ways in which you could attack this problem going forward. And there has been some action in this area, but we certainly have nowhere in any way gone close to solving this problem. Coming back to what I said earlier, where resistance is innate or acquired. When we talk about AMR, we typically talk about acquired resistance. So E. coli, for example, will always be resistant to benzyl penicillin, but that's just the way that is as an organism. That's not what we're so concerned about. What we're concerned about is when an organism like E. coli becomes resistant to amoxicillin by acquired resistance. And as I mentioned, you can have a new mutation in the chromosome of the organism, or you can have transfer resistance genes. And I mentioned before, we have this idea of multi-drug resistance of MDR organisms or superbugs. And what we've seen time and again, every time a new antibiotic or new class of antibiotics even is brought to market, we find evolution of bacteria able to resist their effects. And the spread of resistance outpaces the rate of drug development. And the problem is that many hospital-acquired infections are now resistant to antibiotics, typically multiply resistant to antibiotics. And we're even seeing also problems with community-acquired infections like TB, urinary tract infections, sexually transmitted infections, respiratory tract infections, and so forth. And we can see vertical transmission, so we get a resistance arise in the chromosome. That's just passed on to the daughter cells arising from that particular cell. But more of a problem is that we get horizontal gene transfer, where DNA moves from one cell to another, and particularly bacterial conjugation, where plasmid leads two cells to engage effectively in bacterial sex and transfer DNA. This means that antibiotic resistance genes can often spread very widely, particularly among organisms of the same general group, but even across large phylogenetic distances. I'm not going to go into the detail. You've had talks, I'm sure, about this, about the different mechanisms resistant, but you can modify the drug target, you can modify the cell wall, you can inactivate the drug, or you can have a way of pumping the drug out, or basically the mechanisms in which we can make a cell resistant. So what do we do in terms of microbial genomics, bioinformatics, and AMR? We can we can use these approaches for targeted infection control, and to also engage in better antibiotic usage, better stewardship and usage of antibiotics. We can detect resistant mutations in genes, we can track the spread of MDR pathogens, we can do whole genome sequencing, and as I'll show you at the very end of this talk, we can do clinical metagenomics. And this allows us to also elucidate the mechanisms of resistance, and we can use these approaches. I'm not going to say anything about this, but we can use these approaches to understand the molecular mechanisms and how cells work better. So I'm now going to take you on to my first case study, which is Mycobacterium tuberculosis. Now, tuberculosis is a major burden across the world. HIV has led to a weakening of immune systems and emergence of multi-drug resistant TB, so-called MDR TB, because these patients typically have larger bacterial loads, they're more susceptible to TB, and the challenge therefore is to diagnose and treat MDR TB in order to prevent spread. And genomics has emerged as an important tool here in TB diagnosis and control, in terms of diagnosis and resistance prediction, and in looking at the lineages that are being transmitted, and looking at how what is the major way in which resistance is arising. Is it spontaneously arising, sporadically in each individual patient differently, or are we seeing the spread of multi-drug resistant clones? And if you think about that basically has different ramifications. So if it's basically arising in individual patients sporadically, typically that's because there's poor compliance. They're not taking their drugs properly, and we concentrate on making sure they do that. Or is it that there are clones that are spreading around, in which case we have to try and then target the spread and see if we can find ways to prevent the spread. Here's a paper, fairly recent paper, that basically just reviews the whole landscape of whole genome sequencing in Mycobacterium tuberculosis. So you can use this here to look at surveillance, just basically looking at what's going on around the world. You can look at strain and subspecies typing. You can give you a broad picture of what are the broad strains that are transmitting in different parts of the world and across the world. And there are some strains that have spread all around the world. You can look in at very fine detail at outbreaks, and you can say, that patient there and that patient there are very closely related. That patient gave it to that patient, or vice versa, or they got it in a very tightly defined environment. And you can detect drug susceptibility. So one of the interesting features of Mycobacterium tuberculosis is that drug resistance doesn't arise from horizontal gene transfer. It arises from chromosomal mutations. And so you can actually look at the chromosome and generally fairly confidently predict resistances. So this just shows you what I've been talking about in more detail. Basically, you can take a sample, you can grow it, in TB we tend not to grow on agar plates but on little slopes. You can sequence it, you can map the reads, and you can call out the variants there. Make what we call a SNP table, and you can say a certain position in the genome, the reference strain has this, but the strain in question has a different mutation, and that can be used, as I mentioned, for looking at drug resistance. We can say, that particular mutation there signals that this organism is become resistant to rifampicin, one of the common drugs used. Or we can say, that particular mutation there suggests it belongs to a particular lineage or sublineage of TB, or it's a particular marker of a species. And so you can come up with a sequencing report where you can say it's in a particular cluster, it's got particular resistances, particular strain information. This just shows you the same thing in more detail here, that you can look at all these different mutations, you can predict the phenotypes, and as we're getting more and more information and getting smarter at this, we can actually get even more accurate predictions. And just in the last couple of years now, this has now become totally routine. So the UK Public Health Authorities, UKHSA, now do microbial tuberculosis whole genome sequencing and cluster investigation so routinely they've written a handbook that tells you how they do it. And basically what they show there is you can make different trees, looking at how many variants are shared by different genomes and how many are different. And then you can create what you might call blobograms, where you actually look at those that are all the same and say, okay, so there's one that's one SNP different from those, but all the others are all related, and so forth. And then you can put this on to patients, and you can draw a graph of how this looks in terms of when did those the diagnosis happen, what particular strain was it, and how do the strain emergence of strains map onto time. So I'm just going to very quickly, just to give you a feel for this, take you through analysis that my PhD student Madike Senghor did a few years ago. It's just been written up here. Here's the title and the abstract there. What we looked at was multi-drug resistant tuberculosis in Mali, in West Africa, and particularly in the capital city Bamako. And just to point out what it says here in the abstract, we know that Bamako is faced with the emerging threat of MDR-TB. We performed a whole genome sequencing on 76 isolates, and what we found was there was about 80% of the new cases, 20% were retreatment cases, and about 16% were MDR. And what we found was that most of them belonged to what's called the Euro-American lineage, which is what we'd expect because that's the predominant lineage across the world. But there were genotypes within that lineage, one called the Cameroon lineage, another one called the Ghana genotype. As their name suggests, they're initially identified in West Africa, so there is a kind of geographical stratification to these genotypes. And we found that one particular subclade of the Ghana genotype was an important cause of MDR-TB in Bamako. So if we I just pulled out the figures and tables from that paper there, that just summarises what we found in terms of these lineages. And when we draw a big tree that shows you all of this here, you can see that they fall into a number of very different and discrete lineages, and there are some of these lineages which are actually quite distinct from the lineage 4. So it's certainly not all one thing. There are different strains circulating in those populations. But when you look down at the detail here, you can see, for example, in 4.1, right down at the bottom there, in that kind of blue clade right at the bottom there, they're all the same genotype. They're all this Ghana genotype. Many of them have MDR status, and it looks therefore as if they are actually, we're seeing in that particular case transmission of multi-drug resistance from one patient to another in the community. So we've got a mixed picture here actually. It's not one thing. There is clearly sporadic emergence of resistance in individual lineages and clades, but there is at least one clade where you're still seeing that transmission there. Now this is a moment where I have a bit of a break and I leave you to go off and do this student participation. So if you go to the NCBI website and particularly look at their assembly page, in their search box there you type in Mycobacterium tuberculosis XDR, and then you need to download and unpack a genome assembly. So you just click on the bit where it says download. There's a slight twist in this. If you're using a Mac, don't use Safari but use the Chrome browser. Click on that and you should then download the genome assembly. It may come as a zipped up file. If you go to your downloads folder and double click on that, it should unzip, and then you'll see a series of folders. If you drill down through those folders, you'll get to a genome assembly file, which will be about 4 or 5 megabases in size, and that's the file that you're interested in. What you now need to do is then go to this website here, to which it provides a web interface to a program called ResFinder. So if you go there, and then when it says select your genome, upload the genome that you downloaded to there. What you do have to do is there's an option there to select the organism. Select Mycobacterium tuberculosis as the organism. Submit your file. Wait a few minutes. Go off and have a comfort break, have a cup of tea. Come back in a few minutes' time and you'll see some results. And what I want you to do is just look at those results and get a feel for what we can actually predict and determine once we have a whole genome sequence of a Mycobacterium tuberculosis strain, in this case a so-called XDR strain, extended drug resistant strain. So you go away and do that. I will leave this screen up for a few minutes and say nothing, and then we'll go back to the talk. And at the very end, I will just quickly look at these sites myself, just in case you get stuck. But I'd rather you didn't just scroll right on to the end. You try and do this yourself and get a feel for it. Okay, let's rest here.